The origins of Rhnull blood were never as simple as we thought. For years, the assumption was clean and convenient. A mutation here, a deletion there, and the result was a blood type so rare that fewer than 50 people in all of recorded medical history have ever been confirmed to carry it. That explanation felt complete. It wasn't. What genetics has revealed is more complicated. The reason Rhnull exists reaches down into the architecture of the genome itself, into genes that don't build anything directly but control whether other genes work at all. It is a story about modifier genes, biological switches, and what happens when the switch that controls the switch fails. This video is about what Rhnull actually is, where it comes from genetically, what it means to live with it, and why the science behind it is far stranger than a simple mutation story suggests. By the end, you'll understand why geneticists consider this blood type one of the most instructive anomalies in all of human biology. Before you can understand why Rhnull is so extraordinary, you need to understand what blood types are and why they matter. Your red blood cells are coated in proteins and sugar structures called antigens. These sit on the cell surface like identification tags, read constantly by your immune system. The moment it encounters one, it doesn't recognize. It launches an attack. That's not a flaw. That's exactly what it's designed to do. The most familiar classification is the ABO system. It sorts blood into 4 types based on which antigens are present. Type A carries the A antigen. Type B carries the B antigen. Type AB carries both. Type O carries neither. Your serum also carries antibodies, primed to attack whatever antigen you don't have. Give a Type A person Type B blood, and those antibodies attack the donated cells immediately, clumping them together, blocking vessels, triggering organ failure. This is why blood typing exists, not as bureaucratic formality, but as a biological necessity. The wrong blood in the wrong body isn't just ineffective, it can kill. For most transfusions, matching ABO type and one additional marker gets you close enough to save. But close enough only works when the blood in question is common. When it isn't, the calculation changes entirely. The ABO system is the one most people know, but it is nowhere near the whole picture. The second most important classification is the Rh system, and it is significantly more complicated. Where ABO deals with 2 antigens, the Rh system contains 61 distinct proteins that can be present or absent on your red blood cells, each one a potential trigger for an immune response in the wrong recipient. Most people encounter the Rh system through a single letter at the end of their blood type, the plus or minus after your A, B, AB, or O. That symbol refers to one of those 61 antigens, the D antigen, the most immunologically significant. Carry it and you are Rh positive. Don't and you are Rh negative. Negative blood types are rarer, more in demand, and more carefully managed by blood banks. The D antigen is the most famous member of a much larger family. The other 60 vary across populations, and for most people that variation doesn't matter. The antigens you carry match closely enough with whoever's blood you might receive that no serious immune reaction occurs. The trouble starts at the edges. When someone is missing several Rh antigens, finding a compatible donor becomes genuinely difficult. Medical databases flag these individuals. Blood banks maintain special reserves. The system strains. And then there is Rhnull, which isn't missing a few antigens. It's missing all of them. To be missing one Rh antigen is unusual. To be missing a handful is medically significant. To be missing all 61 is, statistically speaking, almost impossible. And yet it happens. In fewer than 50 confirmed cases across all of recorded medical history, a human being has been born with red blood cells that carry 0 Rh antigens, none of the 61. That is what Rhnull means. The null is not shorthand. It is a precise biological description of an absolute absence. Doctors call it golden blood, not poetic license, but a clinical assessment. Rhnull can be transfused into any patient with a rare combination of missing Rh antigens. For patients with unusual Rh profiles who cannot safely receive standard blood, it is often the only option. Universally compatible within the world of rare Rh blood types in a way nothing else is. But golden implies value, and value implies demand. That is precisely where the problem begins. Fewer than 50 known carriers exist on the planet. Of those, only a small handful are active donors. The blood they produce is, without exaggeration, among the most medically precious biological material on Earth. Rhnull is typically discovered by accident, a routine screen, a prenatal checkup, a standard donation that returns an anomalous result. Nobody is born knowing they have it. The diagnosis arrives quietly, and then it reshapes everything. Here is where the story stops being a medical curiosity and becomes something genuinely remarkable about how genes work. For decades, scientists understood that Rhnull existed, but not fully why. The assumption was straightforward. Something in the genetic code responsible for producing Rh proteins was broken or absent. A simple deletion, a mutation in the right place, and the proteins never got made. That explanation is partially correct, but it's only half the story. Two distinct pathways can produce the same outcome. The first is the amorph type. Here, the genes directly responsible for encoding Rh proteins, the RHCE and RHD genes, are themselves mutated or deleted. The machinery that should build the proteins is damaged at the source. No blueprint, no product. The second pathway is more surprising. It's called the regulator type, and it involves a completely separate gene, RAG, which encodes a protein called the Rh-associated glycoprotein. This protein doesn't build Rh antigens directly. It acts as a chaperone, escorting Rh proteins to the surface of the red blood cell. Without it functioning correctly, the Rh proteins are produced but never delivered. They never reach the cell surface. To any immune system scanning that cell, it is as if they don't exist. This is the modifier gene mechanism, a gene controlling whether another gene's product ever becomes functional. The genome is not a simple list of instructions, but a system of systems, and sometimes the switch that controls the switch is the one that fails. Knowing genetics is one thing. Understanding what it actually means to move through the world with Rhnull blood is another. The Rh antigens most of us carry are not just compatibility markers. They maintain the structural integrity of the red blood cell membrane. When all 61 are absent, that support disappears. The red blood cells of a Rhnull carrier have an abnormal shape and a significantly increased fragility. They break down faster than they should. The result is a mild but chronic hemolytic anemia, a condition where red blood cells are destroyed more quickly than the body can replace them. Fatigue, shortness of breath, and jaundice are the common symptoms, not debilitating for most carriers, but a constant low-level cost. That fragility limits how often a Rhnull carrier can donate. The body needs recovery time. Donating too frequently worsens the anemia, so donors can only give a limited amount each year, making an already scarce resource scarcer still. According to widely reported accounts, one Swiss man with Rhnull blood had his life shaped by it from childhood. Summer camps were off the table. Outdoor sports were avoided. His parents understood that a serious injury without compatible blood could be fatal and planned his life around that risk. As an adult, his solution is methodical and quietly extraordinary. Twice a year, he donates his own blood to himself, building a private reserve for emergencies. He cannot travel to countries without advanced medical infrastructure. The constraints that exist are absolute. Fewer than 50 known carriers, a handful of active donors, blood that expires in roughly 6 weeks under ideal conditions, kept at between 2 and 6 degrees Celsius the entire way. The logistics of golden blood are a masterclass in how fragile a supply chain becomes when the resource at its center is genuinely irreplaceable. When standard options are exhausted, the search goes international almost immediately. Hospitals contact national blood services, who contact each other. The network is small, informal, held together by the willingness of a tiny group of donors across multiple continents. Shipping blood across borders sounds straightforward until you try to do it. Temperature control is non-negotiable, and any break in the cold chain renders the blood unusable. Customs delays are not a bureaucratic inconvenience here. They are a potential death sentence. Some countries maintain highly restrictive policies on blood imports, creating barriers for patients with rare blood types who need urgent transfusions. A package held at a frontier for several days may arrive too late or not at all. The system works, when it works, because individual people make individual decisions to show up. There is a phone call that some Rhnull donors have received, and it is not an easy one to sit with. In one documented case, a Swiss man with Rhnull Blood received word that a newborn needed his blood urgently. His blood was not preferred. I needed it. In the way that it is needed, the alternative is death. He took a taxi to Geneva, took time off work, and donated. Under the donation framework in his region, none of those costs could be reimbursed. The taxi, the lost wages, the disruption, all of it fell on him. He paid, in the most literal sense, to save a life only he could save. That situation raises a question that medicine doesn't have a clean answer to. At what point does biological rarity become a moral obligation? Most of us accept blood donation as a civic virtue, something we should do more of, but understand it as voluntary, a choice. Nobody is on call. For someone with Rhnull blood, that calculus is different. The pool of people who can help is so vanishingly small that each donor carries a weight. Regular donors simply don't. To decline may be the specific reason someone dies. That is not a comfortable thing to ask. And yet the alternative, a world where someone with Rhnull blood in crisis has no one to call, is worse. What rare blood reveals is something common blood obscures. The entire system, every transfusion, every surgery, every soldier pulled from the edge during wartime runs on human willingness. It rests on people deciding someone else's life is worth showing up for. The origins of Rhnull were never a single mutation. They were two entirely distinct biological pathways, one dismantling the machinery at the source, the other pulling the switch that controls whether the machinery runs at all. A reminder that in biology, the simplest explanation is rarely the complete one. But science is only part of this story. The other part is human. Fewer than 50 people carry blood that the rest of the world cannot replicate, navigating lives shaped by a fact they never chose, donating to themselves twice a year, taking taxis to Geneva because a newborn needs what only they can provide. The genome doesn't care about logistics. It doesn't account for customs delays or the cost of a taxi. It simply produces what it produces and leaves people to build a life around it. And medicine, for all its technology, still depends on individuals, on the quiet decision to give something of yourself when someone else has no other option. If this video gave you something to think about, consider subscribing. There is more where this came from. And if you have ever donated blood or been meaning to, today is not a bad day to follow through.